Hi guys, my name is Jesse. You can call me Dr. Brown. Today we're going over the questions of what is the main definition for Calvin cycle, photorespiration, C4 plants. We'll also look at C3 plants. CAM photosynthesis, regeneration of RuBP, reduction of CO2, RuBP carboxylase, CO2 fixation, ATP synthase, and Noncyclic Pathway Photosystem. Okay? So you might ask, what is the Calvin cycle? So simply put, the Calvin cycle, also known as the Calvin-Benson-Bassham CBB cycle, is a series of biochemical reactions that occurs in the stroma of chloroplasts in photosynthesizing cells. It's the primary pathway for carbon fixation in photosynthesis, in which carbon dioxide is converted into organic compounds such as glucose. The cycle is named after Melvin Calvin, who was awarded the Nobel Prize in Chemistry in 1961 for his work on the cycle. The next thing that we're going over is photorespiration. So you might ask, what is photorespiration? Photorespiration is a process that occurs in the leaves of plants, during which oxygen is used instead of carbon dioxide in the reaction catalyzed by the enzyme rubisco. That's ribulose 1,5 biphosphate carboxylase oxygenase. This results in the production of energy-inefficient byproducts and the consumption of ATP and NADPH, which are necessary for photosynthesis. Photorespiration occurs in all plants, but is particularly pronounced in C3 plants. They use a lot of water, which are the plants that use the Calvin cycle for carbon fixation. Then we have C4 plants. So C4 plants are different than C3 plants. They don't require so much water. They can live longer, and a lot of people call them weeds in general. But C4 plants are plants that have evolved a specialized photosynthetic pathway to minimize the photorespiration and maximize carbon dioxide uptake. In this pathway, carbon dioxide is the first fixed in a specialized cell type. It is first fixed in a specialized cell type called a bundle sheath cell, and then transported to the mesophyll cells for use in the Calvin cycle. This allows C4 plants to maintain a higher concentration of carbon dioxide around the rubisco enzyme, reducing the likelihood of oxygenation and increasing the efficiency of photosynthesis. Then we have CAM. So what is CAM? CAM is the Crassulacean acid metabolism. And then we have CAM photosynthesis. So CAM photosynthesis is a great process. It is a specialized photosynthetic pathway that occurs in some succulent plants, such as cacti and pineapples. In this pathway, carbon dioxide is fixed during the night when stomata, small pores on the surface of leaves, are open, and then used during the day for the Calvin cycle. This allows CAM plants to conserve water by reducing the amount of water lost through transpiration during the day. Then we have regeneration of RuBP. And it looks like we might not have enough chance to answer all these. A regeneration of RuBP ribulose 1,5 biphosphate is an essential step in the Calvin cycle in which the 5-carbon compound ribulose 1,5 biphosphate is regenerated from the products of the initial carbon dioxide fixation reaction. This allows the cycle to continue and enables the plant to fix more carbon dioxide. I'll try to get in one more. Reduction of CO2 RuBP is the process, refers to the process of reducing carbon dioxide to a more reduced form, such as glucose, as part of the Calvin cycle. This process requires the use of reducing agents, such as ATP and NADPH, which are produced during the light-dependent reactions of photosynthesis. So we're going over a little bit over 5 minutes here, but we're going to look at RuBP carboxylase. That's ribulose 1,5-biphosphate carboxylase. And this is the enzyme responsible for catalyzing the initial carbon dioxide fixation reaction in the Calvin cycle. It converts carbon dioxide and ribulose 1,5-biphosphate into two molecules of 3-phosphoglycerate. Carbon dioxide fixation, or CO2 fixation, last one, is the process of converting carbon dioxide into a more stable reduced form such as glucose as part of the Calvin cycle. This process is necessary for the synthesis of organic compounds and is essential for the survival of photosynthesizing organisms. Finally, I'm just going to go over ATP synthase, and that's this one. We won't cover this today. ATP synthase is an enzyme that catalyzes the synthesis of ATP, adenosine triphosphate, from ADP, adenosine diphosphate. And inorganic phosphate, ATP, is an important energy-carrying molecule that is used by cells for a variety of processes, including the synthesis of cellular components and the transport of ions and molecules across membranes. ATP synthase is located in the mitochondria and the thylac. So that's it for today. Hope you liked it.